you, James, and thank you, John, and DPUK for inviting me to cross the pond and share this perspective with you guys today. James and Simon really just set the stage for me, and you'll hear I'll be reiterating a number of the points that Simon just discussed. And over the next 15 to 20 minutes, I'm going to share a little bit of background and then 3 stories of how we're filling gaps in Alzheimer's disease. The first will focus on amyloid. The second, my favorite, protein tau. And then the third is not focused on one particular, but calling out that there really is a gap to fill and proposing a framework for us to kind of come together and work towards to accelerate solutions for patients. Okay, so while today I'm going to be sharing with you my own views, they were shaped by my experiences, of course. I'm currently employed by Eisai. I've spent many years at Genentech, but my translational research in Alzheimer's, my career started really with Randy Bateman at WashU. And it was there where I really started understanding what translation meant. And there are different definitions, but in this case, it was truly going to the patient's bedside and bringing those samples back to the bench. And so with this piece in mind, let's start talking. Okay, so we've already, actually, there was a question even raised yesterday about the right questions, and when we think about successful drug development, increasing our odds here, we really need to start working and having biomarkers developed in parallel with any drugs, and we need to be answering these questions at the right time. And while you can take a risk and maybe move a molecule forward without this data, if you want the best chance of success, you are going to answer the right questions. And it takes many, many years, especially if the biomarker may be used someday to help decide a path for a patient diagnosis or what treatment they should get. So I've kind of bucketed biomarkers in a couple different ways. There are multiple types of biomarkers that we use. I'm just kind of simplifying here. In the right drug kind of category, we have those biomarkers that we want to measure over time and see if there are changes. And the couple examples I'll talk about today are the pharmacodynamic biomarkers. A subset of that, of course, is the surrogate biomarkers. This is perhaps the highest bar for a biomarker to reach for validation, is a surrogate biomarker. And then, of course, in the right patient category, we have the one you're most familiar with is diagnostic. But of course, then, as we were talking yesterday, there are risk biomarkers, such as ApoE4 in Alzheimer's. There are prognostic biomarkers, like tau. And there are predictive biomarkers that tell whether or not you'll respond to a drug. Okay, so where are we heading? Where do we collectively want to go, and hopefully not the so distant future? Ideally, you're going to be going, all of us are going to be getting a blood test. And this blood test is going to have a panel of different biomarkers, some of which might be genetic, some of might be protein-based. And they're going to tell us who's at high risk of developing the disease long before symptom onset. And we're going to have multiple options available here, and based on your individual profile and your signature of which biomarkers are abnormal, we'll find the personalized treatment for you. And then in order to know whether those treatments are working, we're going to have a whole bunch of surrogate biomarkers that are going to help us along the way and to predict. Because as you know and you've heard, these diseases are incredibly heterogeneous, and you have multiple drivers and pathologies moving your own pathology forward. So where are we today with biomarkers, as you already have heard, for Alzheimer's disease? Now, first I'll take a step back historically. As you know, Alzheimer's is confirmed at autopsy based on the pathology for amyloid plaques and tau tangles. And as you were just learning, and many of you are well familiar, we've kind of put forward this ATN framework to use in research and clinical trials that has really helped us accelerate by coming together and focusing on the amyloid biomarkers in CSF and imaging, and just generally comment for our field, right? Like CSF and imaging techniques are our window to the brain. So we've been starting there. And so we're most advanced really with the ATN biomarkers, but of course the most excitement, and again 5 years ago I did not think I'd be here saying this, blood biomarkers really are truly emerging. And so it's a very exciting time for us in the field. And so we know through common use of these biomarkers in multiple observational studies, we're placing these biomarkers and the time of the changes in relation to disease onset. And we know in red that it seems that amyloid starts this cascade of events. And so it's no surprise that pharma companies started many years ago coming up with strategies to target amyloid. One of those was an antibody against amyloid. And there have been many agents targeting amyloid that have been in development. And just to put in perspective, kind of simplifying this here, it took us over 20 years to get to the point of having a breakthrough. And that breakthrough was not possible without biomarkers. But again, for the biomarkers, it took us more than 10 years to get amyloid PET to be approved. And it took a while for people to start using it routinely in their clinical trials to select amyloid positive patients. And these amyloid PET tools also ended up being the ways we selected the right dose to move forward into the validation in the big phase 3 trials. So amyloid PET really was key for this success. And that came because everybody started using these tools. We even had to find a way, because there were multiple tracers, the Centiloid project, so we could put them on a common scale and use data, so enable patients greater opportunities to use different amyloid PET tracers. And so these are a few examples of anti-amyloids, adu, gantenerumab, donanemab, that used it in their trials, and there are others, that started establishing the response of this biomarker to treatment. And so in the bottom right is the definition from the FDA of a surrogate endpoint. And I know there is a debate whether or not we've reached this point for amyloid, but I think we are starting to get there. And this is when the biomarker change in response to treatment is reasonably relative to predict the clinical outcome. So I'm just going to show you some of the data that has gone in, and this is from the lecanemab phase 2 trial. And this is an example of data that was showing that a response on amyloid PET seemed to be related to the clinical outcome. And so if I can draw your attention to the purple arrows, so the down arrows is indicating that there is more clinical benefit or improvement, and then from right to left, like greater amyloid clearance. So similarly, from right to left is with increasing dose, so the lower dose being in red and the higher dose being on the left in blue, there seemed to be a relationship here between the dose and the response, and that helped the lecanemab team determine what is the right dose that they wanted to move forward into their phase 3 trials. So we've made great advances, but I told you it took over 20 years for us to get there. And if we're going to get to that vision piece where we have multiple options, we really got to pick up the pace. And so the next, or my favorite target that's most promising because of its relationship to cognitive symptoms, is tau. So I'm going to talk to you about another antibody that is in development for tau. There are different approaches to target tau. But E2814 is an antibody that targets the microtubule binding region of tau. And this region is thought to be really important for the seeding and spreading and propagation of the pathology. And one reason I really like talking about this molecule here is because it was discovered and developed in collaboration between Eisai and UCL, right here with Rohan De Silva. And so recently, the DIAN-2 team, dominantly inherited Alzheimer's network, selected this anti-tau for their new tau arms of their platform trial. And so that study is enrolling dominantly inherited Alzheimer's patients, and kind of coming full circle, Dr. Mummery's site is part of the DIAN-2 network, so this site right here at UCL, and is part of Eisai's phase 1b. It's a very small study looking at the safety of this antibody in patients and giving kind of an early look at some biomarkers. So that's a little bit about the drug and how through collaboration we're hoping to kind of accelerate drug development, more on that right target question. But if we're now talking more about also right target, right patient, we're doing more collaboration, and this time we have a long-standing collaboration with WashU, actually my former lab, Randy Bateman's lab, leveraging their mass spec-based technologies. They really have a nice method that's been developed to target peptides in the MTBR region, and this focus project was taking observational trial samples, so CSF samples from patients in blue that are non-mutation carriers, or yellow or red were the mutation carriers. And so what Kanta Hori, a colleague of mine, found was if he looked at changes over time in these participants of these two peptides, that we can see, if you notice the red trend line, changes and increases in these peptides many years before the onset of disease. And this data helped the joint DIAN-2 and Eisai team, when they were designing the trial, to decide, hey, we're going to enroll asymptomatic and symptomatic patients into this trial. And so now beyond that, there are a lot of different tau biomarkers that are emerging. How are we going to know which ones to develop? Part of it has to do with the question that you're asking about. And the one fruitful thing that's coming from this collaboration with Dr. Bateman's lab is that Eisai and WashU seem to have shared aims in the questions they're trying to answer. And so also by leveraging this technology, they can compare multiple biomarkers on the same platform and in these really well-characterized cohorts, such as BioFinder and these ADRC cohorts, so they can put the biomarkers in context more quickly. And so one example here is the new biomarker MTBR tau 243. And so this particular biomarker has been found to be tracking a bit better with tau pathology as measured by PET, and that in comparison with some of these other phosphocytes, that it has this stronger relationship. And so this helps us think through, which of these candidates should we be pushing forward? And again, it kind of comes down to that question of how we want to use these biomarkers. And so this is one way that we're doing this, and I believe time will tell, but I think we're accelerating our development of tools for tau quite well. But beyond that, there is a big gap to fill. And I think now zooming out, we need more tools. And we need to thinking of that from a translation perspective and from a right questions perspective. And so there are 3 different buckets here to call your attention to. One is that we really need more translational models. So many of the models are really not predictive of things going on in the clinic. And there are a lot of advances that are very exciting. I'll talk about one non-human primate model in a sec. There are reverse translation. We need really to take these clinical biomarkers that we're more routinely using in the clinic back to our non-clinical studies. We need to be looking at plasma NFL, for example, in all of our mouse studies. And we also need to be working together sooner to establish more surrogate biomarkers, because it's such a high bar. And before, you obviously need drugs that are efficacious in doing something, but if we understand the natural history of these new biomarkers, and we're all using the same tools, we're going to be able to implement them faster and answer the question faster. So just a couple examples before I close. Here's one new non-human primate model from John Morrison's lab at UC Davis. And so they're taking an AAV approach to give mutant tau to these monkeys in the entorhinal cortex. And they can see within a few months, they're seeing pathological changes that mimic what's happening in Alzheimer's disease. And so in this type of model, in these large primates, you actually can do imaging, you can do cognitive assessments, which they're qualifying. So if you can imagine, if we were implementing these in early drug development, it might give us a much better chance of translation. The other thing that we need to be doing more of, and we often use these kind of research tools, and they're really variable. We need to use more precise, robust tools sooner so we can be comparing apples to apples. One approach, just quickly, because I got my 1 minute notice, that Roche has put forth is the Neuro Toolkit. And they're a diagnostics company, and so they don't typically have people use their research-grade assays, but they have now opened up this collaboration model, which is really cool, and that different academics, pharma companies are now starting to use these different robust tools. You see some of our favorite biomarkers on there, the classics as well as emerging ones. And I think through using tools like this and sharing data, we're going to be able to more clearly know which of these biomarkers to advance to help populate that ATI and S. And so this is one approach that I encourage us to do more of, as well as more pre-competitive consortia. Simon shared with you some great ones, and here are a few others that are doing important things to help fill gaps. For example, the NIH Biomarkers Consortia has a project now where they're validating a new PET tracer for synaptic markers. The Global Biomarkers Consortia is doing really important work to compare different assays so we know which ones that we should be using. And now with the emergence of the blood biomarkers, how are we going to get that out in common use in everyday practice? So there are working groups moving forward on that. So I believe we really are truly filling these gaps. We've made the most advance in Alzheimer's and amyloid, and we're making a lot of promise in tau. But of course, there's a lot more to do for these other targets, inflammatory targets and whatnot. I think it's through thinking about these translational questions and thinking about the right questions, working together on this, that we are going to make a much more faster progress in finding solutions for dementia. So thank you.
Thank you, Dr. Wildsmith, and perfect timing again, which allows us some time for questions from the floor, so we're happy to take some questions.
Yes.
First question, yes, please, in the middle table here. Can we turn the microphone on? Is there...
Testing.
All right.
Thanks for some great talks. Andy Payne from Lilly. I just was going to, maybe a question for Sir Simon, actually, but please, I would welcome your comments as well, Kristin. Given that the targeting extracellular tau, albeit with sort of passive immunotherapies, to date hasn't been very successful. I'm thinking semirinumab from Roche and Zagotenemab from Lilly.
Yeah.
Why do you think this approach is going to work now?
It's a very good question. As he's alluding to, many of the, what I'll coin the first generation anti-tau antibodies, they all had negative readouts in Alzheimer's disease as well as PSP. And so there are a number of hypotheses about why that is. It could be they weren't targeting the right piece of tau. I think, that's one layer. Maybe the N-terminal part of tau isn't right in the case of a few of them. So I think the field obviously has learned a lot about the target and how the different pathologies, and I think the newer versions are kind of going after more pathological epitopes. That's one hypothesis. Perhaps it's the backbone, so some of the newer ones are not on IgG4, they're IgG1 backbones. So those are just a couple thoughts on that topic. Yeah.
Any further questions? Yes.
Cornelia.
Here down in the front right.
Very nice talk. I really enjoyed it, Cornelia. Can I ask you, what are the risks if you start targeting tau in terms of that you're just simply having a tougher problem because you're late in the process?
Do you mean if we're in the symptomatic space?
Well, even in the propagating phase, that it's getting propagated outside the brain, and therefore it's much more difficult to target. Do you have any evidence, for instance, of the other primates that is more difficult?
I'm not sure I understand.
So if you look at the disease process,
yes, tau comes kind of second.
They are a bit late in the process compared to tackling amyloid. And isn't that a bit of a danger?
Well, I think
I don't say you shouldn't do it, because I think we should.
No, yeah. So I think this is going to be a hot debate in the field, right? Because the reality is, with the emergence of anti-amyloids, will there be a space even for monotherapy? And does that even make sense? And I think there, of course, it's going to be a combination of things. Because of the dynamic range of tau, I think there's space even potentially in moderate AD and later AD for slowing it down. Of course, we all want to be going into the pre-symptomatic space, but I think we're going to have to be evaluating these things together. And until we do the experiment in the clinic, we won't really know. Yeah.
Thank you so much. I'd like to just thank you once again, Dr. Wildsmith.
Thank you.